Hello, everybody. Thanks for the introduction and the invitation to be able to speak. So I'm here. I'm going to be wearing a lot of different hats today. Some of them will be slightly strange hats. But I'm going to be talking to you all about functional genomics, which is why we're all here. I really had to kind of think of ways to follow up on Leah's terrific talk this morning, aware that I'm kind of towards the end of the day, so what can I kind of offer you to kind of keep you interested over the next 15 minutes? Well, obviously there's going to be science. That was a key request from John as my overseer. But I thought, what else I could bring? I can promise you giant microbes. I can promise you Greek warriors. And I can promise you robots. And now that sounds like a great movie for most of us. But I think for me, I'm going to steal this quote from a colleague who's up at Sanger, Ben Lehner, who's also part of this Atlas of Variant Effect. For me, this really kind of emphasizes the problem as I see it, and really kind of underpins everything that we do as a lab. So if you take a tiny little protein, right, 100 amino acids, and you say, how many ways can I kind of create that with the kind of 20-odd variants per amino acid, you're actually looking at more potential combinatorial ways of making that single 100 amino acid protein than there are all the atoms in the universe, right? For me, that's kind of mind-boggling, but it means that there's a lot of stuff for us to work on. And then it gets even more complicated. If Kenny was giving a talk today, he'd tell me that number's pathetic compared to when you're looking at the combinatorial variation across an entire genome. But in a nutshell, this is kind of defines what we do. It's a recently christened field, but the field has been there for some time. It's been called chemoproteomic genomics. Really, all that means is we're taking, oh, Lord. I swear like a pirate, so if I'm controlling myself when I say, oh, Lord, as my team will tell you, no doubt. So we take basically, sweet. I'm not even going to bother. We take information gleaned from protein, typically enriched through chemical proteomic methods, that's the chemoproteo, and then we're using a functional genomics approach to really try and boil into those specific protein features. And what this is really saying is that we're kind of interested in proteins and protein features. We use now a range of different systems. So actually a lot of the methodology we set up was in this kind of green little section, so Toxoplasma gondii. I hate diploid organisms as well, like Leah. So actually the nice thing about Toxo is it's a haploid organism, which makes genome editing really, really easy. And then since then, we've now kind of branched out into other areas. So we're now looking at rare disease in humans, and we're also kind of branching out into other systems. So Leah, I can't see her right now, but Aspergillosis haploid, yeah, it's a good one. Candida auris, another haploid eukaryote. So there's a lot out there for us to kind of have fun with in terms of kind of simple editing, but we're also potentially starting to do some work with bacterial pathogens as well. The particular way that we do this, we kind of, like I said, look at protein features and actually give you some examples. We look at covalently druggable residues, PTMs, and kind of allosterin. I think it'd be useful just to kind of for everyone to think about potential other applications of our functional genomics approaches. So the one I'll focus on first, I swear that he is wearing a fig leaf. So this is Achilles. So for example, one application when we're thinking about functional genomics and kind of trying to understand amino acid function are these disease-associated missense variants, and everyone's been talking about these today. One kind of classic example is actually a kind of oncogenic form of KRAS. So this is KRAS G12C. This particular mutation is associated with about 20% of all lung carcinoma. So it's a highly prevalent mutation. And what I think maybe you don't appreciate is you can kind of think about that mutation in different ways, right? So it's a very specific amino acid change, but you're changing the biochemistry of what's happening on the surface of the protein at that position, such that it's not only pathogenically associated, but we actually know as the switches to a cysteine, we can then actually, it's kind of chemically tractable. And so we can design drugs to try and go in and hit that site. And actually, that's now a target for, I think it's Amgen, Lumacras. So they have a small molecule that covalently engages with that mutation on KRAS. And so you actually have selectivity as a kind of a result of this particular pathogenically associated missense variant. And so one thing we work towards is this idea that every protein has an Achilles heel. And what we can try and do at the systematic level is actually try and then think about this at scale. So now we've had Greek warriors and we've had a giant microbe. So if we're thinking about a bug like MRSA, so if you go onto giantmicrobes.com, you can actually buy MRSA as a big cuddly toy. On a more serious note, we need more targets, right? So you can make the argument that we don't need more molecules. We have pharmaceutical companies with billion-member libraries. People are quite often synthesizing trillion-member libraries with cyclic peptide technologies in single tubes. What we need is better validated targets, and so how can we go about figuring that out? So that we get to a point where we actually have an Achilles heel for every single protein in the proteome, and so then we kind of get to this big vision like was pitched by Leah. You've got different types of atlas. Ours is a vulnerability atlas, where you could kind of go and you could be like, right, we want to hit this protein, and I could tell you where you'd need to hit it and with what kind of molecule. So people do this prospectively in mass spectrometry and with mass spectrometry approaches for broadly in kind of a non-targeted way discovering chemically tractable targets. And so generally the method is such that you take some kind of amino acid selective probe, chemical probe that will kind of selectively engage with an amino acid chemistry on the surface of your proteins. You do this in a protein lysate, and then you use a proteomic technique to figure out which specific proteins are liganded hit by your molecule. And you are able to now do this, it's generally referred to as reactivity profiling. We have a range of different selective probes now, so this is probably about 4 or 5 years ago, there was about 50% of amino acids had some kind of selective chemistry available for them. And so what we can now do is kind of go in and prospectively profile to look for targets of these, but then you end up with these mass spec lists. And so these mass spec lists are great. However, as with all mass spec, if you've ever worked with it, they're very long. So these lists are typically kind of, I don't know, hundreds, thousands of proteins long, and so you're then trying to figure out which ones you want to work with. So you need to be lucky. And I'm not particularly lucky. And ultimately, also, humans are incredibly biased. And so, again, to kind of emphasize this point, if you take all clinically approved drugs that are currently on the market, all of them, we're hitting about, again, this is kind of, the number won't be quite right, about 700 proteins. So that means kind of what, like 99% of the human proteome is not being kind of targeted with a small molecule. So we need to be able to kind of do this in a less biased fashion and stop cherry picking. And take a more systematic approach to really identify function. And so then this is where I engaged in a kind of deep period of thought, where what we realized after 4 years is that we were effectively reinventing the wheel. And that when we were working in our little parasite system, Greg, Finley, Doug Fowler, and everyone else kind of doing the human genetics side of things have been working on this for some time with these saturation mutagenesis, the deep mutational scanning. But I would make the case that there are different types of wheel. At least this is how I justify it to myself. It helps me sleep a little bit better. So this is our approach for kind of targeted proteome engineering. So we take we use a dual CRISPR strategy. Let's see. I'm always terrible at using these things. So start one, right? There we go. We use a dual guide CRISPR strategy, so we target a residue of interest, an amino acid of interest, and we provide the cell with repair templates. So CRISPR is effectively the targeting of the enzyme to that particular point in the DNA is like a fix-me signal, right? The nuclease cuts the DNA, and it's the fix-me signal, and then we tell the cell what to repair it with. And so the cell will then repair, and we provide it with a series of mutations. We provide the synonymous wild type, which allows for a kind of internal calibration of our readout. And then we always put in a stop codon, and we generally work with essential genes. So this will tell us what happens if we break that protein. Those cells are then designed such that one cell will only be mutated once. We then have our kind of assay. Typically, we work with a kind of something simple with a growth readout, but we also have some kind of more advanced assays we're working on at the moment. And then we use that incredibly expensive counting technique that we call next-generation sequencing. And so all you're really doing is counting your relative frequency of different variants in your pre and your post condition, and then you kind of normalize to our synonymous wild type. We know what a broken protein looks like, and so then we can actually quantify individual contribution of individual amino acids to protein function, and we do this in live cells, obviously. The data just give you a sense of how it looks at a kind of raw level and not a heat map level. If we're looking at this protein called ISPH, it's a protein involved in isoprenoid biosynthesis. It has an iron-sulfur cluster that's part of its catalytic mechanism, and that iron-sulfur cluster is coordinated by a bunch of cysteines, so the expectation is if you remove one of these cysteines, it breaks the protein, and so the kind of enzyme shouldn't be functional, and the parasite should die. We can see when we do our transfections with our templates, we can then generate an integration-specific product, so that's one little trick about our method that's slightly different as well. When we go in, we include a synonymously recoded section, flanking, and mutation, and we prime off this in a selective fashion and then outside of our repair template. So it means that it doesn't matter if you've got terrible integration efficiency, it doesn't matter if you've got terrible transfection efficiency, we only look at the data that we want to see. And so again, that kind of really cleans up our data set. So our wheel is slightly different. I'll keep on trying to say that. And then when we actually quantify by NGS, that you kind of see what you would expect, right? So we can normalize to our synonymous position. Stop codon breaks our protein, and our mutations can't kind of rescue because they can't function in the same biochemical way as cysteine does. Whenever we do this again, we've finally got round to the point of doing this with all naturally encoded amino acids now when we go in, and when we're actually kind of doing the rare disease-associated variants, we also include the specific codon associated with the variant itself. And like I said, I mean, I won't dwell on this because Leah gave a terrific talk, but we are, to all intents and purposes, extremely similar to what is kind of being done by our colleagues working with these multiplex studies of variant effects and saturated genome editing. I would point you towards this very, very nice review that was put out by the Atlas of Variant Effects Alliance. I think that was maybe last year or the year before. I've nabbed these pictures because they were just much better than anything I could draw. But I have referenced it, so I think that's okay. I think if you're trying to think about how our approach would compare, it's really our focus, right? We're not looking at individual exons that we then kind of saturate. What we're looking at is specific protein features. So then we're looking across potentially hundreds of thousands of genes of these specific protein features, and we run these assays in parallel. Such that, just again, everyone loves a heat map. This is the kind of data we get. So this is where we were looking at a bunch of different ribosomal subunits. All of these have a reactive cysteine. So these are cysteines that we identified with one of those chemical tractability assessments. So we know we can actually hit these cysteines with a small molecule, and then we can kind of identify different profiles that kind of give us an indication of kind of various sensitivities. And actually, on the basis of this, we then went on and identified a novel small molecule that will only inhibit parasite protein translation and doesn't hit human, and we presume it's acting through engagement with one of these kind of covalently ligandable cysteines. But really, this kind of saturation of amino acids allows you to go explore biochemical space. And PTMs are a really good opportunity to re-pitch that. And actually, this is where I kind of come in with the human functional genomics side. When we're thinking about PTMs, this is, you have your intact protein decorated with chemistry, and then what the cell does is add an entirely new level of complexity on top of that, right? You're adding different PTMs on top. So these are post-translational modifications. There are now more than 400 different PTMs described. So you can imagine how that really complicates your kind of assessment of proteoforms, right? And even, let's say your protein is phosphorylated at 10 positions. Are those 10 positions phosphorylated together all at the same time? Are there some that are mutually exclusive? And we don't really have a way to get at that at all yet. So I think this is a big problem and some kind of challenge that we're going to be coming towards in the future. But ultimately, our functional understanding of these is kind of tiny. So less than 3% of the 250,000 phosphosites have actually got an ascribed function. So PTMs is a kind of nice use case because the addition is chemically selective. So phosphorylation, serine-threonine phosphorylation will only take place on serine and threonines. Likewise, cysteine S-acylation, that will only take place on cysteines, and that's because of the amino acid chemistry. And so then you can kind of go back, and with that in mind, you can then reinterpret your data and think about it in a different way, right? So actually, cysteine 60, we know, is palmitoylated. We're not quite sure what's going on with the aspartate, but again, we think we might be able to then pull out signatures of functional post-translational modifications, and ultimately use this as a way to try and understand protein function. You can imagine you can then layer on top of that information about variants. And so we've now shown that our engineering approach works in human cells, so we use the HAP1 system that Leah talked about this morning. It's a model haploid human cell. What's neat is this cysteine 60, and again, this is kind of crazy, so I find it kind of crazy. My lab think I'm crazy for saying that. This is a cysteine in the ribosome protein in RPS11. It's completely conserved between toxoplasma, and this, so it's a protozoan parasite, right across to humans, completely conserved. Both are palmitoylated, so both are post-translationally modified with this specific lipid modification. And what Kevin's gone on to show is that both are actually completely essential for function as well. So again, this kind of interesting piece of functional information that we're following up on at the moment to really try and understand why this particular PTM on this particular cysteine is so important for the function of this ribosome subunit. What we also noticed in the data from UniProt, actually we just kind of pulled this out, there is an annotated pathogenic variant that sits just proximal to the cysteine. So again, the expectation is that it might be affecting the addition of this PTM. And what's kind of interesting with Kevin's data with the mutagenesis is we actually see that the specific pathogenic mutation that's been mapped, we see it seems to be, or at least in this assay, and we need to refine this assay, we actually see a gain of function effect. So I think that's kind of interesting and really goes to the point that was raised in one of the questions earlier, that we can kind of capture mutational effects in both directions, and we shouldn't really make that kind of a priori decision about how a mutation should be affecting function in relation to kind of pathogenicity. And so then that leads me into really describing what we do very broadly in terms of our project within the Human Functional Genomics Initiative, is to try and annotate variant function, but really taking a very specific mechanistic class of variants. So variants that are sitting at or within a certain distance from a mapped site of serine or threonine phosphorylation, and use that as a way to really kind of provide an immediate mechanistic insight into how these variants are impacting protein function. So if we see that a given variant is, when we in our assay, is again focusing on the VUS, if we can kind of confirm in our engineering that it is actually impacting protein function, then we can kind of immediately go on and test that with our proteomic methods. There's a lot of different moving parts to this. We have a front end with a lot of genetic modeling and computational analysis, right through to the final section when we're then going to be hopefully putting the data back into EBI and using that for immediate use. A couple of little tiny mini vignettes in terms of what we've been producing so far, some kind of cool new tools just to kind of introduce you to. CRISPR generally is not really a gene-centric approach. Sorry, it is a gene-centric approach, but so that's not very useful for protein engineers because of the mapping. So when you're trying to choose your guides to engineer your protein, they're kind of scattered across that intron-exon structure, and so the mapping gets very, very complicated if you're wanting to hit a very specific amino acid site. So Akash, who's actually sat down at the front as well, has developed our new tool. I think we've changed the name now. I think it's Core Designer. But really, it's to simplify this process for protein engineers. You can kind of come in, you put in your UniProt ID, and then it'll really, in specific relation to our approach, it'll provide you guides that will then allow you to target a specific residue that you've identified. It'll provide the guides. It'll then kind of go on, and it'll provide your HDR templates. All of these you can then immediately have pitched with your homology regions attached as well. Such that you can then, again, thanks to our really nice relationship with Twist, and actually to a lot of the conversations we've had with Julian, so I would kind of recommend you go and talk to Julian about developing projects and technologies as well as simply using their products. We can now go directly in and with an onboarded vector, just kind of put in our sequences, and then they ship them to us in transfection-ready DNA, and so that really speeds up our whole workflow. On the other side of things, and I should say actually that should be coming out soon. I think most of the code is up on Git. I'm getting a thumbs up, which is the yes. So I think that's, and again, we'll be putting that on Barchive very soon. Another little quick thing on the modeling side that is a really, really nice announcement for today. This is so new that I haven't even had time to go and access it yet. So Alicia, David at Imperial, a long time ago established a database called Missense 3D. And so what they've now done is kind of do a modified, updated version called Missense 3D PTMDB. And so this is a new database that you guys can now access. It's live. You can access it through this QR code. It's really taking a lot of the data that's available and trying to map it and make it into a more accessible version for really interpreting variant position or known map variant position within protein structure in relation to then your kind of 3D structure and where PTM sits. So to try and understand and visualize in a little bit more detail how these things can potentially interact and how you might be able to interpret variant function through these specific protein features. This is your input page. You can put in your UniProt ID, give you a flavor in terms of how the data then looks when it comes out. You have your kind of query residue position. It shows you all the variants that are being mapped, and then it'll show you these pre-calculated distances. The pre-calculation obviously means it's very kind of light computationally, which means this is extremely fast to use. And I think that really the big thing is the visualization. And so then this is really, really nice. You have your kind of 3D representation, and then because you have your kind of distance matrices, you can actually look in space within this kind of live structure to actually look at where your variant sits, and then where it sits in relation to kind of specific PTM sites, right? So that's your kind of serine residue. Finally, I'll just then talk about kind of where we are in terms of the bigger project overall and how that's going, and we're kind of more or less at the point of kicking this off without it. So the team have been working extremely hard, in addition to all the bioinformatics that we've been doing in the tool development, establishing an automated pipeline, because when we do our mutation, we're looking at individual sites, but per well. So effectively, we do this in an arrayed format. And so what we need to be able to do this is a lot of robots. And so this is really our, part of the collaboration is with the London Biofoundry. And I'm just showing a little video here. Benedict is extremely efficient. He's not actually part of the team. That's his normal working speed. And the robots are just trying to keep up with Benedict. But they've been really kind to share this video just to kind of give you a flavor of a lot of the different kind of instrumentation we have available to us. And so we're now at the point with the pilot where we have, I'm calling it the pilot because we're trying to really flesh out the parameters for the main screen. So the main screen is going to look at 1,000 variants distributed probably across around about 1,000 odd proteins. We're looking at 120 within variants that sit within a specific angstrom radius. So again, we've had to go in, and for all the 90-odd thousand serine, threonine, phosphorus sites, we then mapped those onto all available alpha-fold structures. We then kind of computationally calculated the radius, the 10 angstrom radius, and put that as a distance. We've then gone in and selected out variants within those distances to kind of get our set of pilot sites. We're looking at about 50 or 60 benign and 50 or 60 pathogenic to really get that kind of nice window on our assay to make sure we're able to kind of capture the disease variants that we want to see. And so that's going to be kicking off soon for these 120 variants distributed across about 74 genes. And so then with that, I'm just going to kind of stop and kind of give the acknowledgements and thank really everyone in the team. So this is the team on the left, particularly within the Functional Genomics Project, Kim, Sarah, and Akash. Everyone that's been contributing there as well. So a lot of people who aren't officially badged to the project, a lot of the PhD students have been putting in a huge amount of work. Our team with the functional genomics side of things, I'm just kind of like showcasing their work. And so I think if you're interested in the new Missense DB-PTM, I would kind of suggest you go and have a chat with Alessia. She's here today, as is Ryan. And then just finally, obviously, thanks to the funders. We had a fantastic meeting in Barcelona recently with the team, and thank you very much for your time.